Welcome back to The Deep Dive, a show where we pull back the curtain on the technology that is fundamentally changing how we approach knowledge. Today, we are undertaking a deep dive into the very infrastructure of future scientific discovery. You could almost call it a grand unified theory for how AI talks to robots.
It's a really essential conversation to have. For years now, we've all been hearing these incredible stories about large language models, about specialized AI agents that can reason and hypothesize. They can plan complex experiments, sometimes even better than humans. The potential of this AI scientist idea is it's undeniable.
But that's the thing, right? If the potential is so great, why does scientific progress, outside of a few very highly funded institutions, still feel so bespoke, so one-off? Why is it that when Lab A publishes some amazing result they got with their AI, Lab B can't just, download the workflow and instantly replicate it?
That is the core frustration we need to talk about. It's because the infrastructure just isn't there yet. Right now, most of these advanced AI systems for science are tightly coupled. They're completely customized for a single lab or single platform. They rely on these ad hoc, custom-built interfaces to connect the dry computational world of models and data to the wet physical world of lab instruments.
So if I get this right, if I want to run a complex workflow that needs, say, a high-performance computing cluster for a simulation, and then a specialized database lookup, and then a physical liquid handler in a clean room, those three things just don't naturally speak the same language.
Not at all. It becomes a massive custom-coded integration project every single time.
Yeah.
We're missing the standardized pipes. We're missing the shared operating system that understands and governs scientific context. We lack a lingua franca. And that technological gap is what's holding back true cross-institution collaboration and scalable, reliable reproduction.
Which brings us directly to the focus of this deep dive. The Science Context Protocol, or SCP. This is an open-source standard designed specifically to solve that infrastructure problem by enabling what the researchers behind it are calling a global web of autonomous scientific agents.
The goal here it's audacious. It's to move from these isolated pockets of brilliance to a genuine collective intelligence.
Okay, let's unpack this. Our mission today is to explore exactly how SCP provides this unifying language. We're going to dissect its unique hub-and-spoke architecture. We'll look at the sheer scale of the assets they've already integrated. And most importantly, we're going to examine 4 incredibly detailed case studies that show how it orchestrates everything, from ingesting a simple protocol PDF all the way to running a closed-loop molecular engineering cycle. We're aiming for maximum detail so you can understand not just what SCP is, but really how it works.
Right. We're essentially charting the path from what you might call the scientific stone age of manual scripting and ad hoc integration into a future of truly scalable, reproducible, and autonomous research.
Let's jump right in then. Section 1, the emergence of autonomous science. We've established these systems are already doing extraordinary things. Give us a feel for some of the key early systems that really laid the groundwork here.
Well, we've seen this powerful trajectory toward full research autonomy, and it's been fueled by 2 things: the computational leaps of LLMs, and just as importantly, the physical advancements in lab automation. And these early systems, they really proved the concept was viable.
Let's start on the materials science side. The one that always comes to my mind is ALAB.
ALAB, yes. The Autonomous Materials Synthesis Laboratory, that was a landmark demonstration. It combined computational intelligence with robotic execution to perform these really complex chemical syntheses. What made it so impactful was the sheer continuity and efficiency. Over 17 continuous days of operation, ALAB autonomously searched, planned, and executed. And in that time, it successfully created 41 novel inorganic compounds out of 58 targets.
41 novel discoveries in less than 3 weeks. And the system managed all the decision-making and the physical execution. That's just a staggering rate of discovery.
It really is a rate that very few human teams could ever match. And that capability, it quickly spilled over into the pure chemistry domain. We saw these LLM-based agents like ChemCrow and Cognitus start to emerge. These agents were given predefined sets of tools, which allowed them to autonomously plan these complex multi-step chemical syntheses. Everything from optimizing cross-coupling reactions to synthesizing specialized organocatalysts. They proved that AI could move beyond just retrieving data to making complex physical chemical decisions.
So we're past the point of asking, can AI do the experiment? The answer is clearly yes. Where else have we seen these agents getting traction?
But the paradigm is expanding really rapidly, especially into the life sciences. You have platforms like Orgy integrating sequence analysis, structural biology tools, design algorithms to automate the entire process of designing and engineering functional proteins.
So you could drastically accelerate developing new enzymes or biocatalysts.
Exactly.
Yeah.
And similarly, in environmental and earth science, you've got frameworks like EarthLink. They're demonstrating autonomous reasoning by connecting these highly heterogeneous data sources, climate models, sensor networks, ecological databases, to do complex tasks.
Like predicting regional climate impact.
Right, or assessing ecological stability.
So all of these systems, whether it's something like Intern-Agent orchestrating specialized sub-agents or Origin focusing on a specific biological task, they all point to this future, a future of an interconnected web of scientific intelligence.
Precisely. When you equip them with robust, domain-specific tools, these agents can automate critical research steps and execute entire end-to-end investigations. But, and this is the really critical transition point, these individual breakthroughs, as brilliant as they still suffer from fragmentation.
Which brings us right back to the core limitation that SCP is trying to fix. If we have all these smart agents, what's the actual technological hurdle stopping them from talking to each other today?
The hurdle is the absence of context management at the infrastructure level. The orchestration tools we have now, they only operate at the level of an individual application call. They focus on low-level messaging, call this function. But they completely lack a protocol-level abstraction for the scientific context and for the lifecycle management of the experiment itself.
And scientific context is much more than just the function call. It's all the metadata, the security envelope, the history of the project.
Exactly. Scientific context means providing a persistent, traceable identifier for the entire project. It means tracking the history of decisions made by the planning agent, managing the state of an ongoing experiment. Is the sample currently incubating? Is the simulation halfway done? And crucially, it's about enforcing boundaries, things like data access and permission boundaries across totally disparate platforms. If I use a proprietary model from institution A and then a robot from institution B, the system needs a consistent shared context to manage that collaboration securely and, legally.
And without that structure, any kind of cross-institution or multi-agent collaboration just remains a logistical nightmare. It's hard to ensure reproducibility if the context, the who, what, when, and exactly how, isn't consistently captured and managed by the underlying protocol.
That's a critical distinction. A lot of general-purpose protocols treat interactions as stateless events, just one-off messages. Science, however, is inherently a stateful, longitudinal process. SCP provides that necessary infrastructure layer to turn these isolated breakthroughs into scalable, collaborative, and above all, reproducible science.
So if context is the problem, the only way to solve it is with a whole new blueprint for communication. Let's transition into Section 2, where we can break down that structure. SCP is built on two foundational pillars designed to handle exactly that context and heterogeneity gap.
And these pillars are designed to ensure seamless integration and secure management, which, are the prerequisites for scaling any kind of autonomous science.
Okay, walk us through Pillar 1.
Pillar 1 is unified resource integration. The fundamental objective here is standardization. We need a universal specification for describing and invoking all scientific resources equally, and I really do mean all. This has to span the dry resources, software tools, machine learning models, vast data sets, and the wet resources, the physical instruments, the robotic liquid handlers, the analytical gear in the lab.
So you're standardizing everything from a complex quantum chemistry simulator that's running on a supercomputer all the way down to a simple physical temperature probe. That sounds like a monumental task given the huge diversity in hardware and software out there.
It is a massive undertaking, but the outcome is that you get protocol-level standardization. Once a resource is wrapped in an SCP tool specification, AI agents and applications can seamlessly discover it, call it, and compose its capabilities with others across highly disparate platforms and institutional boundaries. If it speaks the SCP language, it's interoperable. It abstracts away all that vendor lock-in and the need for constant custom coding.
That abstraction is key. Okay, what about Pillar 2?
Pillar 2 is orchestrated experiment lifecycle management. This involves creating a secure service architecture, the hub and spoke model, to manage the complete experiment lifecycle. And that means everything from the initial objective registration and planning through the actual execution and real-time monitoring, all the way to the final archival of the results.
And the authorization piece, because that's paramount for any proprietary research or sensitive data.
Absolutely. The architecture enforces fine-grained authentication and authorization throughout that entire lifecycle. This is what allows for federated collaboration. Institution A can securely share access to its specialized protein model, while Institution B can share access to its data, and it's all managed centrally. This architecture elevates the protocol from just simple tool interaction to the full, regulated, and traceable lifecycle of a scientific experiment.
Okay, let's drill down into the core components, starting with the centralized brain. The SCP Hub. Now, if the hub is the central orchestrator, that sounds like it could be a massive single point of failure. How does SCP distribute load or ensure uptime across potentially hundreds of labs?
That's a critical challenge in any distributed architecture, you're right. While the hub is the central authority for context, governance, and planning, the actual execution and the data storage are federated across the SCP servers. So the hub isn't running the simulations itself or moving the robot arms, it's managing the plan. And for fault tolerance, the hub itself is designed with redundant distributed services. But its key function is authority. It maintains that global registry of all tools, datasets, agents, and instruments, and it handles service discovery, task dispatch, tracking, and governance.
So let's say an AI scientist or a human researcher submits a high-level request, something like synthesize this compound and characterize its thermal stability. How does the hub turn that very open-ended instruction into an executable plan?
That process is handled by what's called the intelligent orchestration layer. The hub uses advanced AI-driven intent analysis models, often LLMs that are fine-tuned on scientific reasoning, to translate that natural language objective into a set of candidate task graphs. And these graphs are multi-step experimental plans that involve specific tool calls, data retrievals, wet operations, and computational models.
And it doesn't just produce one plan, it gives you options. You mentioned it ranks the top K executable plans. This is where that scientific travel agent analogy comes in, isn't it?
Exactly. The hub is the ultimate meta-planner. It doesn't just pick the first viable path, it evaluates those candidates against the current environment in real time. It checks instrument availability, current resource budgets, required input data, and all the user permissions. So for each experiment, it might rank, say, the top 3 to 5 executable plans.
And it provides the decision rationale, the pros and cons for each of those plans.
Precisely. Plan A might be fast, but requires access to the proprietary synchrotron imaging server, resulting in a high-cost estimate. Plan B could be slower, but uses only open-source computational tools and locally available lab equipment, offering the lowest risk and cost. The hub surfaces the dependency structure, the expected latency, the estimated cost, and the projected experimental risk for each option. This lets the human or the high-level AI client make a really prioritized, contextual decision.
That level of pre-planning is incredibly powerful, and this planning system is tightly integrated with the AI governance module you mentioned. How does that module actually operate on the back end? I mean, what data is it using to detect conflicts?
The governance module's job is to ensure that the proposed scientific activity is safe, feasible, and compliant. It uses several real-time data streams to do this. First, it tracks instrument reservation logs and real-time sensor load reported by the SCP servers. So if Plan A requires 3 hours of continuous use of a specific mass spectrometer that's already booked for another critical job, the module flags a resource conflict, and the hub can then deprioritize Plan A.
So it's performing real-time resource forecasting and capacity planning.
Correct. It also monitors budget constraints and checks security permissions. If a proposed workflow involves combining highly toxic reagents that are tracked by an internal inventory system, the governance module can flag that risk level.
Yeah.
It doesn't just issue a binary, yes or no block. It can issue early warnings, suggest alternative, less hazardous routes, or maybe suggest delaying the execution until the required resources or the budget are available. It's the intelligent gatekeeper for the whole platform.
Okay, so once a plan is selected, the hub compiles it into that crucial element, the fine-grained, repeatable, protocol-level workflow specification. This structured JSON task graph. Why is this specific JSON structure so much better for reproducibility than, say, just a detailed methodology section in a scientific paper?
Because the JSON task graph is the canonical machine-interpretable contract for the experiment. A traditional narrative protocol is written in ambiguous human language. Even with the best intentions, it leaves room for interpretation. Add solvent slowly until solution turns cloudy. The JSON protocol, however, it forces complete explicit traceability.
Can you give us a hypothetical example of what that structure actually enforces?
Certainly. Each node in that JSON graph is a single, discrete atomic operation. It will explicitly contain the globally unique tool identifier, like Thothop Centrifuge V 2.1. It'll have the precise input parameters, like RPM 5000 duration, 600 seconds. It defines the expected output schema, like a structured data file containing mass and temperature readings. And critically, it has explicit dependencies, like Node 3A must complete successfully before Node 4B can begin. The JSON forces the capture out every single parameter needed to replicate the action exactly, and it serves as the persistent audit record that's versioned and stored by the hub.
That makes perfect sense. The JSON file itself becomes the executable contract, guaranteeing that what was planned is fully traceable, auditable, and repeatable by another machine. So after the planning phase, the hub manages the automated execution, feedback, and interaction. What does that look like in practice?
The hub dispatches each step, strictly following that dependency order from the JSON graph to the appropriate SCP server. It tracks the task states in real-time running, paused, completed, failed, and it logs server response latency. But the real sophistication is in the validation and feedback loop.
And what about that critical rollback feature? You mentioned the hub can optionally roll back prior actions. That sounds like a really complex feature for wet lab autonomy. I mean, how do you roll back a physical action like heating a sample or mixing two reagents?
That's a great challenge to raise. The concept of rollback in a physical environment is very different from a software transaction. In many computational steps, like a simulation that crashes, the rollback is simple. You just discard the partial result and restart. In the wet lab, as you say, you can't unmix chemicals.
Right, the entropy has already increased.
Exactly. So for wet lab steps, the rollback feature relies heavily on the SCP server reporting precise state changes in sensor readings. If the hub detects an anomaly, for example, the sensor reading from the heating bath server reports a temperature far outside the allowed range. The hub triggers exception handling. It can pause all the downstream steps that depend on that failed heating step, which prevents a cascading failure.
It stops the system from wasting materials on an already ruined intermediate sample.
It's more than that. The intelligent rollback policy might dictate that the protocol needs to discard the failed sample, log the precise failure, and then prompt the planning agent to revise the protocol starting from the previous safe checkpoint. Maybe that means reobtaining the starting region from storage. So, while it can't physically reverse the action, it manages the logical recovery and state remediation, it prevents the failure from propagating, and logs the entire decision chain of that recovery effort. This robust coordination is what really sets it apart from just simple sequential scripting.
That distinction, logical recovery versus physical reversal, is vital for understanding how these autonomous wet lab systems can function responsibly. Now, let's look at the other side of the equation. The SCP servers. These are the edge nodes.
The servers are the vital bridge to the physical world. They manage the local scientific resources, the actual lab equipment, the specialized local databases, the domain-specific models within a given environment, be it a university lab or a corporate data center.
You mentioned that SCP requires standardizing device drivers across every major vendor. That feels like a huge practical hurdle. Is the SCP community building custom wrappers for every single instrument ever made?
That is the core difficulty in adopting any standardization protocol in a diverse hardware landscape. The SCP approach is twofold. First, it defines a standard specification schema for capabilities. So the server registers its device, let's say a centrifuge, based on what it does, like spin sample or set PM not who manufactured it. Second, yes, the community or the institution running the server often has to build these thin, standardized SCP wrappers or adapters around the vendor's proprietary API or driver stack.
So the complexity is contained at the server level. It's the server's job to translate the universal SCP command into the manufacturer's specific jargon.
Precisely. The server shields the hub and the planning agents from all that messy device-specific detail. It receives the high-level protocol command from the hub, translates it into the exact sequence of low-level device operations, the actual moving of a robotic arm, the API called to the specific model instance, and then it streams the intermediate states and final results back to the hub as structured SCP-compliant messages. They also perform continuous health monitoring, reporting device status and resource utilization, which feeds directly into the hub's global scheduling and governance decisions.
And finally, the SCP clients. This is the interface layer for the human researchers and the high-level AI agents.
The client is the gateway. It handles the initial user authentication, making sure the user or agent has the necessary project context and permissions. It retrieves the list of visible tools, filtered by those permissions, and provides the surface for interaction. A human researcher might use a graphical interface to build a workflow or submit a request via natural language. An AI agent just uses the programmatic API.
And since every action from that client carries the authenticated user's credentials and the persistent experiment identifier, the auditable provenance is maintained centrally by the hub, even when the experiment spans 3 different institutions and 5 different tool types.
That centralization of provenance and security is the feature that really unlocks these multi-institution heterogeneous workflows. It brings together remote HPC clusters, literature retrieval services, and physical robotic systems all under one common, enforceable, and auditable policy framework.
That is a remarkably robust framework. Now that we understand the architecture, the brain, the hub, the translation layer, the servers, and the interface, the clients, let's look at the immense scale the system has already achieved in Section 3, because this proves that SCP is not just theoretical.
Absolutely. The Science Context Protocol has already been deployed and tested within the Intern-Discovery platform, and it's integrated over 1,600 interoperable tool assets. This is a mature, massive ecosystem.
Over 1,600 tools all speaking the same language.
Yeah.
That is transformative. What kind of impact does that sheer scale have on the scientific discovery process?
It fundamentally expands the action space and the problem-solving capacity of the scientific agents. Earlier systems were limited to maybe, 10 or 20 specialized tools. Now, an AI can perform complex cross-domain reasoning, combining structural biology, computational physics, and wet lab execution in a single standardized executable plan. It creates a really robust, general-purpose foundation for scalable scientific intelligence.
Okay, let's break down the composition of those 1,600 tools, starting with the disciplinary distribution. Where is the deepest concentration of these integrated capabilities?
It reflects global research investment and the maturity of automation technologies, really. The single largest focus area is biology and related technologies. It accounts for 45.9% of the integrated tools. This covers the entire spectrum: genomics, proteomics, drug discovery, metabolic engineering.
Nearly half of all the integrated tools are dedicated to life sciences. What follows that?
Next up is physics at 21.1%. This includes optics, electromagnetism, fluid dynamics, and condensed matter simulations. Then comes chemistry at 11.6%, which integrates computational chemistry, reaction pathway optimization, and molecular dynamics.
So those 3 core disciplines, biology, physics, and chemistry, really dominate the ecosystem, which shows a focus on empirical and computational science.
Yes, and the remaining tools cover mechanics and material science, 8.7%, which is crucial for industrial and engineering applications, mathematics, 8.0%, which provides foundational analytical and statistical capabilities, and then information science, 4.7%, which covers data management and specialized computing utilities. It's a truly cross-disciplinary foundation.
Now let's look at the functional categories, because this is where the system's abilities really become clear. The 2 largest categories, computational tools, 39.1%, and model services, 13.3%, they're often confused. How does SCP distinguish between them?
That distinction is vital for a technical audience. Computational tools, at nearly 40%, are generally deterministic, established algorithms and utilities. Think about data cleaning, file format conversion, like converting a CSV to JSON, running standard statistical tests, or executing an energy minimization routine based on classical force fields. They perform standardized operations with predictable outcomes.
And model services.
Those are the predictive, learned, often non-deterministic AI and machine learning endpoints. They take an input and they produce a prediction based on a trained statistical model. This includes things like ADMET prediction for toxicity, predicting protein folding structure using an LLM like ESM-Fold, or predicting material degradation rates. The output is an inference, often with an associated confidence score. SCP makes sure both types of resources are callable and composable using the exact same interface.
That clarity helps us understand how the agents partition their work. Deterministic handling of data versus probabilistic prediction. What about the rest of the tools?
Well, the second largest category is databases at 33.8%. This focuses purely on massive, standardized data access. And this isn't just a web scraper. This is highly curated access to repositories like Uniprot, Interpro, PDB, NCBI, and CatG. It turns massive scientific corpora into structured, callable data services for the AI agents.
So we have computation, prediction, and data access making up the bulk of the dry side of things.
Correct. Then you have the crucial connection to the physical world. Wet lab operations at 7.7%. This is the direct control layer for physical equipment. And finally, Literature search tools make up 6.1%, integrating sources like PubMed and specialized literature search engines, allowing agents to ground their planning in published research.
Okay, let's make this connection to the wet lab really concrete. We talked about the ThothOps server, the dedicated wet lab execution engine. It exposes 58 stable atomic commands. Why 58? What does that standardization look like beyond just the generic terms like pipetting and mixing?
The 58 commands represent the minimal, non-overlapping set of fundamental actions that are required to orchestrate the vast majority of standard wet lab protocols in chemistry and biology. And they are stable because they represent universal physics, not vendor specifics.
Give us a specific illustrative example.
Okay, consider temperature control. An instrument vendor's API might require this complex sequence of proprietary messages to set a block temperature. Initializing the heating element, setting PID loop parameters, confirming sensor registration. Under SCP, an agent calls one atomic command, something like set temperature, blockade, target temp, unit duration sec. The 58 commands ensure the planning agent never has to know the messy details of whether it's talking to a Thermo Fisher or a Hamilton instrument. It just speaks a universal language of scientific action. This abstraction is what allows the protocols to be universally executable.
That really defines the power of the standardization. We also saw specialized tools mentioned, like drugs day tool and drugs day model.
That pair illustrates the seamless integration beautifully. The tool bundles the deterministic cheminformatics libraries, OpenBabel, RDKit, BioPython, for things like generating 3D molecular structures or calculating descriptors. The model service handles the predictive side, things like AD-MET toxicity and docking affinity prediction. An agent just sees them as two distinct specialized tools, but SCP coordinates them in a single fluid workflow.
And the materials and fracture analysis tool shows the same kind of depth in engineering.
Exactly. That single server exposes 107 discrete tools for stress strain, fracture criteria, and failure analysis calculations. If an AI agent is optimizing a polymer, it can invoke any of those 107 functions via the standardized SCP interface without needing to learn 107 different APIs. This level of standardized integration is the key prerequisite for the case studies we're about to explore in Section 4.
These case studies are where the theory really meets the road. They truly demonstrate the power of the SCP Hub as an orchestrator, unifying databases, AI planning models, and physical robots under a single unified context.
Right, this is where we see how SCP translates these high-level human goals into structured, machine-executable protocols.
Okay, let's start with case study 1. A researcher submits a high-level objective, for instance. Design and run a PCR protocol to verify a specific gene knockout.
So the Hub receives this natural language query, and it immediately orchestrates the planning. It routes the request to a specialized planning agent, like the Thoth server, which is typically an LLM that's been fine-tuned specifically on millions of successful scientific protocols. The agent processes the intent, it understands the constraints, verifying a knockout, and it begins to generate the steps.
And the crucial step here is standardizing the output of that agent.
Precisely. The planning agent translates its high-level reasoning into a detailed, structured SCP JSON protocol. This protocol precisely describes every single step, which specific DNA primers are needed, the required reagent quantities, the thermal cycler timing parameters, and the expected analysis endpoints. It's machine-readable, and it's guaranteed to conform to the execution standards of the platform.
Once that canonical JSON is generated, the execution can begin.
The Hub forwards that standardized protocol to the execution node, the ThothOP server. ThothOP then decomposes the high-level steps defined in the JSON-like prepare reaction mix into those 58 atomic operations we discussed. It generates the specific device-level command sequences. For example, a precise instruction for the robotic pipetting system. Dispense 1.5 microliters of primer A from well C4 into well A1 of plate 96-2.
And the Hub coordinates the physical dispatch of that command.
Yes. The Hub dispatches these atomic commands sequentially to the appropriate robotic platforms and analytical instruments. The pipetting robot, the PCR thermocycler. It monitors acknowledgments and sensor readings, managing the entire execution cycle. The profound takeaway here is that the user skips the manual scripting, the error-prone protocol documentation, and the coordination effort entirely. The SCP system handles intent recognition, expert planning, protocol generation, physical execution, all seamlessly.
Case study 2 tackles what is maybe the single greatest source of friction in reproducibility today. turning these arbitrary narrative lab documents like a method section in a journal article into executable code.
This is a massive problem. So a researcher uploads an existing protocol PDF. It's written in narrative English, it uses inconsistent notation, and it assumes a great deal of implicit human knowledge. It is the absolute antithesis of machine readability.
So how does SCP ingest that kind of unstructured text?
The hub classifies the task as protocol ingestion, and it routes the PDF to the Thoth server, which now acts as the protocol understanding node. Thoth uses its language model capabilities to parse the free-form text, systematically extracting every structured detail: the objective, the specific materials and their sources, the actions like centrifugation or incubation, the precise parameters, 5 minutes, 37 degrees, and all the dependencies between steps.
And the output is that ubiquitous machine-readable JSON protocol object.
Exactly. The JSON output is a precise enumeration of the workflow, which overcomes all the ambiguity of human language. And critically, once generated, this protocol goes to the ThothOP execution node for planning. And ThothOP doesn't just blindly trust the PDF's instructions, it validates all the extracted parameters against the capabilities and the safety limits of the available registered instruments before any execution is initiated.
That validation step is the safety harness. It prevents the system from blindly executing a step that could damage the hardware or create a hazard. That seems crucial when you're running historical or third-party protocols.
Exactly. If the protocol calls for a volume that the available robot can't handle, or a temperature the instrument isn't rated for, the hub flags the conflict and requests a revision, or it aborts the sequence. The result is the automatic, safe, and traceable reproduction of an experiment that originated from an unstructured document, demonstrating end-to-end control and rigor.
Okay, let's shift gears to case study 3, a complex, entirely dry-side workflow, AI-driven molecular screening and docking. This workflow showcases the sheer composability of those 1,600 integrated tools across multiple scientific disciplines.
This workflow starts with a large list, say 50 small molecules, identified by their SMILE strings. The goal is to filter them down to viable drug candidates, a process that used to take months of specialized labor.
The power of SCP is in chaining these specialized tools together. How does the system handle that initial filtering?
The workflow begins with parallel filtering, which is a big cost-saving strategy. The hub coordinates 2 simultaneous calculations using standardized SCP computational tool calls. First, the Calculate Drug Chemistry tool evaluates the quantitative estimate of drug likeness, or QED. This is a fast, deterministic calculation. Second, the Pre-Molecular YIBET tool, a specialized AI model service, predicts the LD50 toxicity metric.
And why perform that QED evaluation before doing the much more expensive docking and structural analysis?
Efficiency. QED, which scores molecules based on factors like molecular weight, lipophilicity, and complexity, is a strong, very fast predictor of druggability. If a molecule scores poorly on QED, say below 0.6, or presents a high toxicity risk with an LD50 below 3.0, we just eliminate it immediately. In this case, the initial 50 molecules were filtered down to only 6 viable candidates in a matter of seconds, avoiding the massive computational expense of running structural biology on 44 non-starters.
So that initial filtering stage is handled by standardized SCP tool calls over the entire input list. What happens next? Stage 3 is preparing the protein target for the actual docking simulation.
This is a major pain point that SCP just abstracts away. You start with the target PDB ID. Let's use 6EKV as an example. Preparing a protein involves a sequence of specialized, often non-intuitive structural biology tools. The SCP toolchain runs a sequential series of automated operations, downloading the raw PDB structure via one tool call, extracting the target chain, and then a tool like PDB Fixer is invoked.
And what does PDB Fixer actually do?
PDB Fixer is crucial because raw protein structures often have missing atoms, missing residues, or connectivity errors. They're messy. PDB Fixer repairs those structural gaps. This used to require manual intervention just a few years ago. Once the structure is repaired, another tool, UFPocket, is called to identify the binding pockets, the cavities where a drug molecule might fit. The hub automatically selects the highest scoring pocket to define the active site coordinates.
So the user doesn't need to be an expert in structural crystallography software. They just tell the hub, prepare the protein.
Exactly. And the hub translates that into a multi-step error-corrected sequence of tool calls. Stages 4 and 5 prepare the structures and perform the docking. Both the 6 filtered ligands and the receptor are automatically converted into the PDB-QT format, which is what most docking engines require, using more specialized cheminformatics tools.
Then comes the central computational task, the docking itself.
The hub invokes the quick molecule docking tool, feeding it the PDBQT files and the precise coordinates of that identified pocket center. The system runs the docking simulation, and the results, the predicted binding affinities, are screamed back to the hub in a structured format.
And the final filtering stage ensures only the best hits are flagged for follow-up.
The pipeline filters the results based on binding affinity. In this scenario, only compounds achieving an affinity score of less than or equal to -7.0 kilocamol are retained as high-potential hits. The entire process, from 50 raw input strings to 2 highly prioritized hits bound to a prepared protein target, is an automated, traceable, and composable pipeline, managed entirely by the SCP Hub using a diverse range of tool categories. This just dramatically accelerates the early, expensive stages of drug discovery.
Our final case study brings us to the tightest possible coupling of the dry and wet components, AI-assisted fluorescent protein engineering. This demonstrates the true closed-loop scenario.
This is really the state-of-the-art in autonomous science, where the AI system not only plans the experiment, but iteratively and automatically adjusts based on real-time physical feedback. The process starts on the dry side, with the researcher defining a high-level optimization goal. Engineer a variant of a fluorescent protein with, say, 20% higher photostability.
And the hub orchestrates that design phase.
The SCP Hub coordinates numerous computational servers running advanced AI tools for sequence design, mutational scanning, 3D structure modeling, and molecular dynamics simulations. The AI agents explore billions of potential amino acid variants against the defined criteria. The output of this intensive computational search is a ranked list of candidate variants and, crucially, a standardized SCP experiment plan that is encoded for direct use by the wet lab components.
So the dry side finishes and the wet side starts up seamlessly.
Exactly. The same standardized SCP plan is automatically translated into executable lab protocols for the physical construction of the plasmid DNA, the transformation into cells, the cell culture, and finally, the quantitative fluorescence characterization. These steps are compiled into atomic operation sequences for robotic platforms like liquid handlers and plate readers.
And here is where the loop closes, and the system gets truly autonomous. How does the execution feed back into the planning?
This is the core strength of SCP's context management. During the physical execution, quantitative fluorescence measurements, brightness, yields, stability are generated by the plate readers, and they stream directly back to the hub, immediately updating the shared SCP experiment context in real time. An intelligent monitoring agent within the hub is watching this incoming data stream.
And what would trigger an adjustment?
If the agent detects that the yield of a specific variant is unexpectedly low, or the measured brightness is below the predicted threshold, the agent can suggest on-the-fly adjustments. For instance, it might dynamically suggest trying an alternative buffer composition or increasing the incubation time by 30 minutes. The agent then initiates a revised set of physical steps for the next iteration of the physical experiment, all without human intervention.
So the system is running a hypothesis test-adjust cycle continuously, fusing AI-based design with robotic execution into one unified iterative pipeline.
Precisely. SCP is the essential orchestration layer that ensures every single step of this iterative process, from the initial computational design variant to the final physical reading, is traceable, auditable, and contextualized within the larger project goal. It moves science past these sequential stages and into a realm of unified autonomous optimization.
These case studies really underscore that the architecture has to be far more sophisticated than just a simple messaging standard between models. And that brings us to Section 5. The comparative analysis of SCP versus the closest existing concept, the Model Context Protocol, or MCP.
Right. The Model Context Protocol, MCP, is a general-purpose standard. It's widely adopted for integrating LLMs with software APIs and data sources. It standardizes basic interactions, things like function calls. SCP builds on this foundation, but it introduces 3 key extensions that are absolutely non-negotiable for real-world, high-stakes scientific use: the centralized hub orchestration, the generalization of tools to include lab devices, and the explicit modeling of the experiment lifecycle.
Okay, let's look at the philosophical difference first.
Whatever.
Protocol standardization. MCP is message-centric and stateless. It's like a point-to-point phone call. Why is that not enough for complex science?
Because science is a long-term project, not a single interaction. MCP standardizes the syntax of the interaction, but it lacks a structured format for a complete scientific protocol. When different research teams use MCP, they end up layering their own conventions on top to manage the overall project goals, which leads inevitably to fragmentation. There's no universal format for objectives, parameters, and expected outcomes.
And SCP addresses this by providing that higher-level grammar.
Yes, SCP provides a standardized research workflow representation. It turns low-level messaging into a machine-interpretable script for the entire experiment. It uses that structured, JSON-based schema, which assigns a persistent experiment ID. It defines the type, dry, wet, or hybrid, and it tracks every single decision. It transforms a one-off request into a fully contextualized longitudinal project history.
The second area is high-throughput experimentation. If MCP is stateless, coordinating a massive batch of, say, 1,000 parallel experiments sounds impossible without building some massive external tracking system on top.
It is a huge bottleneck. Under MCP, coordinating batches requires external logic and external state tracking, because each tool invocation is, by default, context agnostic. MCP lacks the native concepts of an experiment queue or any built-in state memory across runs.
So how does SCP elegantly fix that for large-scale screening?
SCP treats the entire workflow as a first-class object. A researcher submits a single JSON plan that details 100 or 1,000 trials. The SCP Hub recognizes this as a single experiment ID with many trial variants. The Hub then automatically handles the batch scheduling, manages concurrency across multiple SCP servers, and crucially, it aggregates all 1,000 results into a uniform, standardized format that's linked back to the master experiment context. This just streamlines large-scale screening exponentially.
Moving to the third point, multi-agent coordination. MCP connects one client to one server. But as we saw in the case studies, modern science requires multiple specialized AI agents working together in a structured team.
MCP only defines point-to-point communication. It's like a phone system. It's great for one-to-one conversations, but has no inherent specification for conducting a coordinated meeting or directing an orchestra. If multiple agents need to collaborate, say an agent specialized in literature review needs to hand off a result to an agent specialized in chemical synthesis, you need auxiliary protocols layered on top of MCP just for the agents to talk to each other productively.
And the SCP Hub, then, is the missing conductor managing the conversation flow.
Exactly. The SCP Hub is the centralized coordinator. The agent's actions are always contextualized within a shared managed state. The Hub receives the overall goal. It reliably decomposes it into subtasks like Agent A run the simulation, Agent B prepare the wet lab reagents, it tracks the dependencies, and it sequences the execution. It transforms a series of independent conversations into a coordinated, traceable scientific narrative.
And finally, the most defining difference for hard science: wet lab integration. We discussed how MCP was just not built for physical hardware.
That's the critical divergence. If you tried to use pure MCP for a lab protocol, you'd have to create these cumbersome custom adapters for every new device. And even then, there's no universal standard for lab actions. An AI agent using MCP might output a text protocol for a human to carry out the wet lab steps, because the protocol itself doesn't speak directly to the machinery.
Which introduces massive friction, human error, and completely breaks that ideal of autonomous, reproducible science.
SCP solved this by making wet lab integration a first-class objective. That was Pillar 1. It defined standardized device drivers and vendor-agnostic interfaces specifically for laboratory equipment. The AI agent calls the high-level atomic action incubate at 37 degrees C for 30 minutes, and the SCP middleware and the local edge server handle the translation into the specific manufacturer-dependent commands required by the actual instrument. This universal command vocabulary is what allows AI agents to reliably control physical processes on a global scale.
So, by adding centralized orchestration, persistent scientific context, robust lifecycle management, and explicit support for physical devices, SCP transforms a general tool call protocol into the foundational infrastructure that is necessary for scalable, reproducible, and truly autonomous scientific discovery.
It is the unifying open-source standard connecting the fragmented world of scientific resources, those 1,600-plus tools, models, databases, and physical instruments, into one interoperable, collaborative network.
And the SCP Hub acts as the secure, intelligent brain, managing everything from the initial design intent through resource conflict resolution to the final execution and archival via those traceable JSON protocols. The sheer depth of the sources we've reviewed today, with explicit tool lists covering thousands of specialized functions, really underlines just how comprehensive this effort is.
And the significance for you, the listener, is that this paradigm shift makes complex research reproducible, traceable, and highly scalable. It shifts the burden of coordination, sequencing, safety, and integration from the human or the high-level AI scientist to the protocol infrastructure itself. Researchers and agents can now focus entirely on the why and the what of discovery, rather than the manual how of coordination and integration. That acceleration factor just cannot be overstated.
That is truly foundational. If we can bypass the weeks or months of integration and scripting needed to link computation with robotic execution, scientific progress is going to accelerate in ways that are frankly hard to grasp right now.
And that leads us to our final provocative thought. If the Science Context Protocol succeeds in making complex, dry-wet research a globally accessible, machine-executable API, something you can simply plug into from anywhere, what new scientific discipline, one that today relies heavily on fragmented, specialized, and isolated efforts, will be the first to transition entirely to a fully automated, multi-institution, agent-driven model? What happens when a global research question can be answered not by assembling a consortium of researchers, but by submitting a single fully specified JSON file to the web?